So I'll talk about cold water corals, rather different to the other talks we've had so far today. And what I'm going to do is just give a quick overview to you today in 10 or 15 minutes of what we know about them and what we don't know. Then I want to show you a short 5-minute film, and we're actually kind of premiering this film here in Australia. It's a film that we made with David Attenborough, and we're absolutely delighted that David Attenborough agreed to work with us on this short film to help raise awareness of deep-sea corals. So the talk's going to review what we know about these things. They're very complex structures, just as tropical corals are. I'll talk about the depth of the aragonite saturation horizon in the oceans as a real master variable in controlling their global distribution. A little on what we mean by deep-sea coral reefs and coral carbonate mounds. A little bit of work on food supply and ecology in these areas, really kind of fundamental work that's been coming into place in the last 5 years or so. And then some of the perspectives. What the title of this symposium clearly is Corals in a Changing Environment. What we know about that changing environment in deeper waters, and what perspectives we have in our research, and also looking forward into studies of transatlantic and transoceanic cold water coral research. So to begin with a little bit of history, this goes back a long way. It goes back to the days of Linnaeus. Here is the Right Reverend Eric Pontoppidan in Bergen, who was a correspondent of Linnaeus, and you can see from his quote there's an interesting kind of link there, even to biotech. They were beginning to wonder is there something in this stuff for us? And in fact, he noticed the apothecaries in Bergen selling these coral branches to hang around the neck. If any of you are suffering from plague or apoplexy, come and see me later, and we'll try to continue those studies. But some of the really fundamental work really began with the global voyage of HMS Challenger, the famous Challenger expedition of the 1870s, led by Charles Wyville Thomson. He wrote up his experiences in this book, The Depths of the Sea. This is alongside, of course, the famous multi-volume series of reports from Challenger. The species that I'm going to focus on today is Lophelia petusa, or as it was known then, Lophelia prolifera. Here's a lovely engraving from that book. And this is the species that forms the really significant and large reef frameworks, not only in the Atlantic, but further afield. So coming on to that, here is the distribution that Ove mentioned. So these things are extremely widespread, and all we've plotted here are the known distributions. This is back in 2006, of Lophelia, Madrepora, which is a rather more delicate secondary framework former, and Selenosmilia, which is an important species especially around New Zealand. And you can see this concentration along the Atlantic margins, right up into the Arctic Circle, and right down into the Southern Ocean, and right through the deep water tropics. So very significant distributions, but you can also see gaps in the distribution. I'll talk about the gap in the Pacific in some detail, but the gap in the Indian Ocean is very likely just a sampling artifact. And we're still at that stage. There's tremendous gaps in our knowledge. The North Atlantic is no doubt very suitable by its chemical, the makeup of the chemical oceanography there, a very deep aragonite saturation horizon. But other areas that are very suitable, such as the Indian Ocean, have barely been researched. So let's look at this area just to begin with, and I'll show you a few pictures to give you an idea of these areas. This is Lophelia growing here at the Mingulay Reef Complex, which is the area that I work on off Scotland. That's an inshore cold water coral reef complex at around 130 to 180 meters water depth. Off Norway, these areas are very, very well developed with extensive reef complexes, some 13 or 14 kilometers in length, growing along chains where icebergs have ploughed and scoured along the continental shelf. They can be important fish habitats. This is a sebastes, a redfish, which is often found egg-bearing and gravid in the coral areas, more so than outside the coral areas. But they are also extremely beautiful and interesting areas. These are some of the gorgonians that grow in the Norwegian areas. This is Paragorgia arborea. Coming down now to why the corals are growing, where they're growing. This is a plot from John Ginnott's paper in 2006, where he plotted out the distribution of cold water corals here as green triangles against the depth of the aragonite saturation horizon. And just take away from this the fact that in the North Atlantic you can see a very high concentration here of green triangles, many, many records in that area where the depth of the aragonite saturation horizon, the depth at which aragonite is going to start to dissolve, is around 2,000 to 3,000 meters water depth. Contrast that with the North Pacific, but we find very, very few green triangles and a very shallow aragonite saturation horizon. If we look at that in terms of distribution here of these scleractinian corals plotted in red, you can see that in this area along the Aleutian Island chain, there are simply no records of these corals, but the ecology is filled instead by calcitic organisms. Here we have a variety of calcitic hydrocorals and gorgonian corals, and bryozoans. Here is another redfish, another kind of sebastes, sitting amongst a gorgonian coral in the Aleutian Island chain. Now, further to the south, distant from the actual scouring influence of these icebergs, have grown up very large features that we call now coral carbonate mounds. So I'll take you to this area off the southwest of Ireland, which is probably the best studied area now in the world for carbonate mound development. And this is the flank of a coral carbonate mound that we photographed in 2003 using the French ROV Victor 6000. This picture is taken at around about 800 meters water depth, and it shows the sloping flank of one of these mounds, and you can see the framework corals coming out from that slope, the gorgonian sea fans, the white creatures that you can see are glass sponges here where the arrow is. That's a species, a genus Afrocalistis, that's very common on these carbonate mounds. Also on the carbonate mounds in this area we find other kinds of corals. This is an Antipatherian, a black coral. We don't know exactly what the species is. We think the genus is Leopathes, which is being revised currently. But this is the genus which Brendan Rourke dated off Hawaii, off a seamount there. He sampled this coral, and by carefully working his way through the stem at the base, through the trunk-like stem, he worked out that that individual on Hawaii was over 4,000 years old when he sampled it, which led to a rather funny cartoon that somebody published where the quote was, "Scientists find 4,000-year-old coral and kill it," because that's exactly what they had to do to bring this thing back to the surface. Okay, so coral carbonate mounds, I want to go into those in a little more detail. The area that we're looking at here is the Porcupine Sea Bight, southwest of Ireland. A lot of these dots here do correspond to those mound areas. They tend to form in provinces or clusters. And here's a multi-beam echo sounder map. So for those not so familiar with multi-beam echo sounders, this is a three-dimensional bathymetry of the seabed produced with a swath of beams, about 118 beams of sound, creating a picture of the seafloor. It both gives you bathymetry but also a backscatter record telling you something about the texture of the seafloor. You can see how the mounds tend to be aligned sometimes. They form these clusters, and we have been very interested in trying to work out what's been going on here. They can be 200 to 300 meters in height across the surrounding sea floor. So the immediate questions that spring to mind, what are the accumulation rates? What's controlling that? Is there coral at the base as well as through to the summit? There was a lot of interest 5 to 10 years ago in whether or not they were initiated and nucleated upon gas seepage areas, and even that the corals might be in some sense given their food and nutrition from a chemosynthetic source. So here we have a simple schematic where we go around an early interglacial period where corals begin to colonize. You can see these little stick-like corals coming in. That will set up a cycle of reef development, of reef framework development, as that live reef is degraded and bioeroded. Things that we heard about earlier today, of course, are really significant and important in deep water, but we almost have no data about those rates, about those processes in deep water settings. Over time, coral frameworks develop, but in glacial times, we hypothesized that the areas were inundated and dropstones came down and they were smothered, only to begin growing again when conditions improved in the next interglacial period. And this model has been tested and examined by a lot of people over the last 5 to 10 years, and it culminated in 2005 when the International Ocean Drilling Program drilled through one of these coral carbonate mounds. It was a Challenger mound, interestingly, and from top to bottom they found a sequence of coral reef frameworks interspersed with glacial deposits, hemipelagics and dropstones, more reef framework, and so on and so on. The dating is still being worked out, but at the moment the estimate to the base of that mound was 2 million years before the present day. So these are very significant and very long-lived features, and in this area, southwest of Ireland, we've seen repeated coral reef growth at depth way back 2 million years before the present day in these interglacial pulses. So that's become a very active area around the world as people try to work out what's promoting deep water coral growth in different settings. So that longevity gives rise to, of course, other areas of work, and I'm just very quickly going to skim across these areas. In some places off Africa, for instance, out of the reach of glacial action, there's a good evidence for 50,000 years and more of continuous coral growth. The carbonate mounds themselves can be hundreds of thousands to millions of years from when they began their genesis. We see this incredible importance of glacial history and mound growth over time, and they're beginning now to provide a very interesting paleoclimatic archive. And we can kind of summarize how this goes in a simple flowchart. There's a variety of techniques, and I won't go into these in any detail, but we have some of the pioneers of this work here that have looked at different proxies and elemental proxies within the coral skeleton to try to derive temperature data. These might be magnesium-calcium ratios, strontium isotopes. There's a variety of things that have been used. Stable isotopes of oxygen have also been used. And this is a really very current and very hotly debated aspect of this kind of work. But of course, if you want to try to produce an archive of this time, of this sort, you need a timeline, you need a chronology. And here lead 210 dating has been used, and there are good results from some apparently annually banded Gorgonian and bamboo corals. But that chronology is, of course, so important. But going to the other side of the diagram, the corals have been incredibly useful in estimating ocean ventilation history in intermediate water masses. By parallel dating with carbon-14 and uranium-thorium, we can work out the age of the seawater the coral is calcified from. And in a very nice series of papers in the late 90s, Jess Adkins and Augusto Manghini showed that some corals were even, apparently by their C14 ages, older at the growing tip, the young tip, than they were at the older base. And the interpretation there is the water mass has shoaled, exposing the coral to older carbon as it's calcified later in its life. I hope that makes some kind of sense. But you get the absolute date from uranium-thorium and the seawater DIC date from the carbon-14. A few other archives that have been used. I mentioned the banding chronologies. The kinds of techniques that are used are high-resolution laser ablation and ion microprobe techniques. But as well as individual mounds being incredibly long-lived, some of these corals themselves, like the Gerardia corals and the Leopathes that was over 4,000 years old, provide single-spot high-resolution archives of deep water mass history. So now a little bit about what we know about the animal and how the animal functions in its environment. And I want to take you to the west coast of Scotland here. The area is off Mingulay. You can see the black dot there on the right map. And we'll look at that area in a bit more detail. There were historic records of coral occurrence going back to 1905, but nobody had mapped the area, and we didn't know whether or not any kind of reef complexes or mounds would be found. So we mapped it in 2003 with one of these multi-beam echo sounders. Here you can see rocky high points, channels within. This is exaggerated 6 times, this plot, to make things show up. In this area, you can see clusters of mounds, and all of these areas are individual coral reef mounds. The mounds can be up to 5 meters from the surrounding seabed, and they're all formed by Lophelia. But how do you study an environment like that? You can't go out daily very easily to look at it, and this is a massive block in the work that we do. So at Mingulay, we teamed up with the Dutch Netherlands Institute for Sea Research to use deep-sea landers, which can be deployed on the seabed to record the environmental conditions. We use conductivity, temperature, depth profiles to study the tidal signals and the changes across those reef areas. And we found some really interesting features of the hydrography that help us understand why the corals are growing where they are. And I've got a plot here that I'll just talk you through. I'm going to start this one at the bottom, where we have water temperature on this axis, and the other axis is the same. It's current speed. And you can see that every 6 hours there's a tidal change, very clear. The corals there are exposed to the semi-diurnal tide. But interestingly, at this change of the tide, there's a warming signal, a very clear warming, and again, slightly less, and again. But as that warming signal hit the corals, the fluorescence on the reefs also pulsed as well. And after a lot of head-scratching and thinking and talk to physicists, we worked out that this was a phenomenon called a hydraulic jump, whereby an internal wave moving across this complicated rocky topography is suddenly caused to overbreak and push warm surface waters down across the reef. And every 6 hours, these corals are receiving a warmer, more fluorescent, we think, food-rich supply of food. So we then started to follow up on the ecophysiology of the corals to try to figure out what that means to them and to their basic biology. And again, doing this kind of work is often based on quite small samples and an awful lot of effort. People often ask how we go about getting these things, and we don't have the best technology. Here we're using a video grab to grab those corals and bring them back to the laboratory. They're very sensitive to temperature changes. You can see Q10 values of up to 8, so their metabolic rate increases very rapidly over temperature. We can see here between 6 degrees and 11 degrees, a 3-fold change in metabolic rate for Lophelia sampled at Mingulay. In terms of ocean acidification, we can see here the predicted effects of the contraction and the shallowing of the aragonite saturation horizon over time. And this is the stark message from the modelers. This model is based on Jim Orr's work. And if this is true, then areas that have been suitable for coldwater coral growth for very many hundreds of thousands of years, if not longer, could become inhospitable. Now at this point, I'd like to dim the lights if I could and swap to a short video, and the video is going to give you some moving images and maybe a bit more of a sense of what these areas are like.
And the very word coral probably brings to your mind a picture of sparkling clear water. Waters in a lagoon with a coral reef towards the ocean, and inland a lovely beach of sand with palm trees waving in the gentle oceanic breezes. Well, a lot of corals are like that, but this coral comes from the dark waters 400 feet down, 150 meters deep, off the Scottish island of Mingulay. The word coral conjures up images of blue tropical waters, not the dark depths of the deep sea. But in fact, over half the coral species we know today are found in such waters, not on shallow tropical reefs. Lophelia.org is about these coldwater corals, the habitats they form and the threats they face. Lophelia is the world's most widespread and important coldwater coral, and such corals gradually form huge deep-sea colonies and reefs. Only in the last few years have we discovered that these reefs are found across the globe from north to south, taking thousands or even millions of years to form. The reefs in turn create a rich habitat for many other species, from simple filter-feeding sponges to predatory sharks and other fish. In fact, these deep-water corals have been known for a long time. This is a book by Philip Goss, one of the great Victorian naturalists, about the corals around the British Isles. And here is a picture with exactly the coral I showed you before, Lophelia. And in this book, he describes a whole group of corals that are found around the British Isles. And it's a book that was largely forgotten for a very long time, until now, when we start research again on deep-water corals, and we find that Philip Goss knew quite a lot about them. But we're only beginning to understand this basic ecology. Observing cold-water corals requires long-ranging voyages and advanced technology. Lophelia.org also provides exciting information on these methods, as well as case studies of some of the cold-water coral habitats around the world that we have been able to study.
That's a first. That's an absolute first.
Yeah.
Fabulous.
As our knowledge grows of this previously hidden world, the extent of man's destruction of it becomes clearer. Modern deep-water trawling methods have become increasingly devastating to cold-water coral reefs. The trade in coral jewellery, new developments in deep-water mining, and the potential catastrophic deep-sea oil leaks present further dangers. Corals only thrive in narrow temperature ranges and rely on the chemical composition of the seas to do so. It's these characteristics that leave them vulnerable to their greatest threat of all. The carbon dioxide that man releases in rising quantities has not only increased ocean temperatures, but is also making them more acidic. It becomes ever more difficult for corals in deep, cold waters to form calcium carbonate skeletons in an increasingly acidic environment. All of this means an uncertain future for a world of wonder that we are only just beginning to reveal. These and other issues are all described in lophelia.org, alongside links and news from Coldwater coral researchers across the world. Enjoy your visit, and please tell others to visit the site to learn more about Coldwater corals. As more people learn about these vulnerable species, we can work together to ensure that they're conserved for generations to come.
Okay, thank you very much. Something like recruiting today to spruce up.